So thank you very much for being here today. And today I'm going to tell you a bit about this research, which came out with it's overlapping between my postdoc at in University and my new position at Coach University, where I'm starting my independent. And today I'm going to tell you a bit about how to build hydrodynamic theory to describe it today. So, sure. So, first of all, as would be that some of you are not as familiar, with the output system, I will review a bit some results about the productive fluids mostly. And then I will describe how the paradigm of active fluid could be exploited to give and to build an hydrodynamic description, of course, given description for the dynamics of epithelial. And eventually, I will close the presentation with an example, a material example of how we can try to decipher the dynamics of epithelial tissue flow in air channel, which is a very relevant biological problem of cells usually flow. So, first of all, what is active matter? Active matter is a peculiar state of matter where the individual constituents are able to convert some internal energy into motion. So they can move autonomously. And examples range from human traffic on the scales of hundreds of thousands of meters up to the sub-cellular scales, where instead, like examples are indeed cellular tissues or even sub-cellular filaments. And today I will be mostly concerned with these two final examples, which are tissues, and the image that you're seeing here is a system suspension of microtubules. So microtubules are one of the main constituents of the skeleton of a cell, and these are basically rods-like stiff molecules. But when assembled together, they can be extracted from a cell. They can be suspended at the interface between water and oil, and these would eventually tend to align with each other, giving rise to this kind of pattern that you see here. And furthermore, you can disperse ATP in the mixture, and ATP can be considered as a practically affect the fuel of cells. And what happens is that filaments start to slide with respect to each other, giving rise to these peculiar dynamics. And this was a very famous movie. It's more or less 10 years now. So basically, the idea is that how can we describe this system? Well, I would say that this is probably one of the biggest success of active matter, because the theory that I'm going to describe works very well to describe the dynamics that I just showed you. And the idea is that we can build on the observation of nematic objects. These objects tend to align with each other. So the idea is that I can define a preferential orientation, alignment of orientation, local orientation, so which is defined by this n vector, and build an object which is symmetric under the inversion of x, because these objects don't have a polarity, don't have a direction. They are symmetric under the inversion. So I need an object which has explicit nematic symmetry. And at this point, this is actually an object which is used, customarily used in the physics of, for describing liquid crystals. So I could borrow the equation of liquid crystal to describe the dynamics of this tensor. And the idea is that basically this consists of two sets of equations. One is the Beres-Hilliard equation, which describes the evolution of the two tensors, and the second one is the Navier-Stokes equation. So in the Beres-Hilliard equation, here we have the evolution of the two tensors. So this is object here is the material derivative of my two tensor. This is basically defined by a coupling with the flow. Here I have a co-rotational derivative, this term is telling me how the two tensor evolves under a rotational flow, and where omega is the vorticity of the flow, namely the antisymmetric part of the driving velocity. Here I have a term which is the flow alignment term, and the psi parameter determines the orientation of the liquid crystal molecules when the liquid crystal is under a shear flow. So if I put my liquid crystal under shear, the molecules of the liquid crystal will tend to tilt with respect to the flow. Finally, this term is a relaxation term, which is a topographic elastic term, and would, in absence of any activity, drive the liquid crystal towards a relaxed state where the molecules are all aligned. Furthermore, liquid crystalline systems are fluidic systems, therefore I need to describe their hydrodynamics, and this is eventually can be done with an Navier-Stokes equation, where I have some pressure contribution which enforce the incompressibility of the fluid, a viscous contribution for dissipation, and then a stress tensor which would include elastic contribution. So this is the term which would flow, which would source a flow due to the deformation of the liquid crystal molecules, and this is a flow alignment term, which is somehow the conjugate with respect to the flow alignment. Basically, this stress would source the flow that tend that push the liquid crystal toward the liquid direction. So the coupling between the equations. Yes, basically here there is two. Here there is. Now the point is that this is a passive theory, meaning that can be derived from equilibrium considerations, and however, and this is what we usually use to describe a liquid crystal, classically. However, what I want to do is actually describe an active system. So what I will do is try to introduce a non-equilibrium stress, which describe the force which is sourced by the action of these active agents. And what I can think is that each rod, each element, each active element actually exert a force at the head of the tail. There is no head at the two extremities. And I can imagine that I'm actually in a larger system, so I can cross grain this singular force, and we'll need a better description of this later. And I find out that the stress exerted by a suspension of many constituents can be actually expressed as alpha, which is a phenomenological parameter proportional to the intensity of the force exerted by these active agents and the two tensors. So if I do this and I simulate the dynamic, or I just simulate these two equations, this is the resulting dynamics, which, as you see, very well reproduces the dynamics of a previous experimental movie that we showed you. And I would like to draw your attention on these features of the liquid crystal, which are exhibited by the liquid. So a classical liquid crystal would relax into an aligned state. However, what happens in the active counterpart is that activity is large enough to bend the liquid crystal, bringing it into an intrinsic non-equilibrium steady state, meaning that this situation is at equilibrium. If I measure hydrodynamic quantities, the kinetic energy, the entropy, these objects are constant. However, what happens is that the activity is large enough to drive in bending instability, generating these, for instance, when a deformation becomes strong enough, this would lead to the generation of topological defects. Topological defects are actually points where it's not mathematically possible to define a local orientation of the crystal molecules, and can be classified according to the number of times and the direction the director field wins around the defect itself. So, for instance, this is a positive defect of charge 1/2, as the director wins all the time around the topological defect, while instead it is a defect charge 1/2, but negative, as the director wins in the opposite direction with respect to the point around the defects. And okay, so this was an introduction regarding active matter, but what I want to do now is try to convince you that epithelial tissues or cellular tissues in general do actually behave like fluids, and there is no better way than showing this phenomenologically. What you're going to see now is the embryogenetic process of the fly, the Drosophila megakarnogaster. So what happens is that at a certain point, there is a vertical flow of cells, and the vortex will generate eventually the invagination of this cellular monolayer, generating the three body parts, which will become the head, the abdomen, and the tail of the animal. Same kind of cells, still epithelial cells, but this time are mast cells on a substrate, and what happens is that if you initialize them in a volume, this will undergo instability because cells realize that they are not confluent. There is an edge, and edge in the language of the cells means a wall. So cells are trying to cover and reproduce to tile all the available space. And what I see when I see this movie is actually the same process as wetting a drop of water or oil, wetting a surface. And the final, and these, for instance, these events are processes which are functional of life, bonding, embryogenesis. However, epithelial cells also play a very important role in life-threatening processes like cancer metastatic, the process of metastatic state. This is a movie of cancer cells in vivo in a mouse, which are intravasating into the intracellular membrane and invading surrounding organs. And what I see here is basically a total practical effect, a shear flow of the cancer cells. Now, the idea at this point was, how can we, again, try to describe from a first-grain perspective, so from an hydrodynamics perspective, the dynamics of epithelial tissues. Is this possible? And the first steps in this direction probably date back to 2017, when this article claimed that, demonstrated that topological defects in epithelia govern cell death and extrusion. So what happens is that the cell, cells in tissue, they die, will continuously regenerate. But cells also die, and they need to be extruded from the tissue. So how is this process carried out? And what happens is that as the cell which is dying migrates to the position of a topological defect, and there is extruded from the tissue. Now, what is new in this sentence is the fact that I can define topological defects in an epithelial tissue. Topological defects are something which can be observed in an ordered system. So this means that there must be some kind of order. And the suggestion is that the kind of order in epithelial tissue is actually nematic. So the same kind as the one that I showed you before, even if it is not as clear as striking as the one that I showed you before.
Is there any reason why it could be just polar? It shouldn't be polar. For example, in the embryo, there are gradients which have been laid down. Is it an approximation that you go start with, or
I didn't start it. No, but yeah, it's an approximation. It's you do data analysis, and you
there's no polar, there's no 1/2 defects.
Exactly.
See the 1/2 defects.
But I'm going to discuss this in a second because it's actually, I'm going to go deeper on this. But the point is that we need to define a concept of polarity as well. I mean, I'm very ready to accept the critic, yes. In principle, no, I mean, a constructive critic. I'm trying to answer because it's a methodological approach, and I should consider polar symmetry, which, however, is something that it's not been considered, and there are some reasons that I'm gonna explain later. Actually, even more, there could be other kind of symmetries. And this is going to become clear in a couple of minutes. So the idea that tissue could be nematic objects became actually very fascinating. And you can actually find the literature a lot of articles which actually use the concept of nematic effect in tissue. Now, on the other hand, in the evidence of nematic order in epithelial tissue fostered the proliferation of numerical models for epithelial. And one of the most diffuse is probably the Voronoi model, where you're basically describing each cell as a Voronoi polygon. Basically, the center of better, the centroid of the Voronoi polygon follows an overdamped equation. So where VC is the velocity of the cells, imagine that all cells move with a constant velocity, but their direction actually evolves up. So the direction of the velocity of each cell follows, subject to random increments, and so that the cell itself follows a persistent random mode. The term that you see here is the relaxation of an energy, which can be built in terms of the areas of the single polygons and their perimeters. And eventually, in what we can say is that we can build a dimensional number with this P0, which is called shape factor, which is given by the ratio between the perimeter and the square root of the area. And basically what we find is that a P0 value, which is very small, would actually favor the formation in the system and relaxation in polygons with many edges. While instead, if I consider high values for the shape factor, I would actually start to generate polygons like triangles, squares, pentagons. So what happens is that if I also include activity, so if I increase the velocity, the nominal velocity of each cell, the picture is the following, with the system transition from a liquid state, which is characterized by a lot of dislocations and discrimination in the system, which is so at all practical isotropic, and so I'm allowed to call it liquid, to a solid state. But one transition between the solid and the isotropic state, what happens is that the system passes through an intermediate state, which is characterized by the fact that positional order of the cells is lost, but instead there is a notion of nematic of orientational order that I can still define, and this is hexatic order. So there is an intermediate state where the position of the cells, the positional correlation decay exponentially, but the orientation of the polygon in fact decays with a power. And this is a fingerprint of quasi-numerical Jordan or in other words, liquid crystal. Not nematic, because the symmetry of the space is six-fold symmetry. Therefore, it's not a nematic, it's an hexatic liquid crystal. So this means that the phase is invariant eventually under rotation of 2 by 6. There are other models for epithelial tissues, numerical models. This is based on a phase field approach where each cell could be described as a phase field, I can still again describe the evolution of these cells with an advection diffusion equation. And the advector would actually feature a non-equilibrium velocity, which is somehow a direction which the cells want to follow, and with a direction with the follows random intervals. And if you do this and you put together a lot of the cells, you obtain this kind of system where basically you have each cell which is interacting with their neighbors through repulsive interaction and filling the space as epithelial cells. So even in this case, this model features a transition between a liquid and a solid phase, and eventually you understand that the kind of order that is broken while going between the solid and liquid phase is actually hexatic. So there was this big discussion with this somehow the experimental community was speaking of nematic order and epithelia, while instead the evidence of the order in numerical models for epithelial tissues was actually suggesting the existence of an hexatic symmetry. And this was basically the question that we wanted to address when joining Luca Flores in Leiden. Are tissues hexatic or not? And for this, we used a lot of microscopy features from Julia Egger, which is what at the time was a PhD student in Leiden. Now she's at the University of Queensland in Australia. And so the kind of data that we had are basically pictures of epithelium. So what you do with these pictures, you first pick. You need to proceed with cell segmentation. So you need to define the vertices of the cells, and eventually, now the point is that how do I define a direction? So now I have a polygon, and I want to attach to this polygon a direction. Eventually, I can do this for the case of nematic order with a tensor. So this would be the ellipsoid, the quansoid ellipsoid, the inertia tensor for a system on a given point. And the principal directions of this tensor would give me the main direction of alignment of it. So if I do this, I can actually set the organization procedure, and this object being totally symmetric in 2D and traceless in these two brackets like the traceless, only have two independent points, and which actually can be cast into a complex number with a magnitude and a name and a text, where the magnitude would tell me how elongated is the point, and the phase would tell me in which direction. So now this is more or less what people have done so far, and the point is that we want to test how these how this order parameter responds by testing some defective structure whose charge is known. What you're seeing here is a positive discrimination in an hexatic tissue, and this is a negative discrimination. And if I can basically compute my order parameter for each cell, and the result is that in the first case I get a vortex, which is a topological defective charge +1, and in the other case I get an aster, which is again a topological defective charge +1. So this object, this nematic order parameter, is not matching either the value it can't, it does not have the symmetry to give me a defective charge 1/6, as the minimum charge that this object can generate is 1/2, and it's not even matching the sign of the defects. So the idea is that we can generalize, and what we did was generalizing the shape tensor. So by constructing a tensor which has the right symmetry to capture a generic p-adic symmetry. So the idea is that I want to compare the nematic symmetry with respect to the triadic one, tetratic, pentatic, hexatic, heptatic, whatever, of course. And what you can do is actually define an object where now I don't have the tensor product of the position of the vertex twice, but p times. Now, in general, this is a monstrous of algebraic objects, but luckily, this object is a symmetric under the inversion of any pair of indices. It is traceless, so no matter how you construct the indices, it will give you 0. And luckily, again, it still associates two independent numbers, which are, again, can be interpreted as a magnitude and as an orientation. And here I'm going to focus only on the nematic and hexatic symmetry, but this is a procedure that you can do in general. And so, for instance, you have your pentagon or your whatever, your generic polygon. You can copy this object and you can associate the direction. And in the first case, it would be a rod, but for the hexatic symmetry, right now, the hexatic order parameter should be invariant under rotation of 2π/6. So it has to be a star. Or if you want, the nematic rod, it's nothing else but a star with 2 legs, which is invariant under rotation of 2π/6. And so now we want to test how this procedure responds to to this new order parameter. And what we find is actually that it correctly determines the position and the charge of the topological defect. To see this, you can follow one of the legs by going around the center of the defect, or in the other case, we arrive that the rotation of the hexatic leg would go in the opposite direction with respect to the direction of the contour, which tells you that the charge is negative. So we were pretty happy with this tool. How can we proceed? So basically, right now we have a tool to define, to put a rod or a star on top of a cell. But this tells us nothing about the symmetry of the system. To see what is the symmetry of the system, I need to focus on one point and see if the symmetry stays in space or it decays or how it decays. So what you do is that you can basically fix the position, you can average, you can constrain your order parameter and radius, and eventually to understand, to have some insight about the kind of order that you have in your system, you can look at how the order, the magnitude, decays over space. As I enlarge the averaging radius, I, of course, will see the magnitude of the nematic order going down, and it goes down with the power law, which suggests the presence of quasi-long range order, or eventually a nematic difference. So eventually we can redo this operation. For the case of hexatic, we can define the position of the stars. We can constrain the star, and eventually we observe again a power law decay for hexatic symmetry. However, what we observe is that hexatic symmetry is larger at the small scales, and there is a crossover point where instead over which nematic symmetry becomes strong. So the idea is that basically, if I need both an hexatic description at small scales with permanent epithelial tissues and a nematic. And
is it conceivable there are other symmetries?
Absolutely yes. But I can. It's totally correct. It's absolutely conceivable. But we actually tested this, and I can say, with the statistical trustability that these are the two main. So in the range where hexatic symmetry is larger, hexatic symmetry is the largest one statistically. Well, instead, at the at the larger scale where nematic symmetry is dominant, this is the largest one with respect to area, other symmetry.
So what exactly is the measure for? You say that you compare with.
I mean, the two-factor for the distribution. So we collect a point, and we are doing we have a collection of data points, and this collection of data points, so we measure P for P equal 1, 2, 3, etc. And we observe that where the symmetry is larger, we can say that it is statistically large.
So you measure all the moments Q
up to 7, 8. I mean, for sure, 7 is on the. So, and the idea is that if you look at this again at this movie, what you could think is that nematic and hexatic symmetry is the one which governs the dynamics of cell-cell interaction. But instead, there is another scale that you can identify in this movie, and is the scale of the compression of the cellular layer, which are the darker region in the system, which actually set up a totally different scale and would eventually be responsible, at least this is the idea, of the migration of cells on larger scale. So, building on this observation, we wanted to introduce hydrodynamic theory. So,
maybe I missed this. What experiment is this? What kind of?
So these are NDCK cells, and built on substrate. So it's an in vitro experiment, and yeah, it's a flat substrate, and
boundary conditions.
Well, you grow the tissue, and then you're imaging one part, the center. So you wait some time. There is no boundary, or at least the boundary is so large, I mean, so far away that you can basically.
They grow from the center and they.
Well, that's not exactly. I mean, yes, but these are basically strains of cells. So I've seen this procedure many times. So usually you need to wash your cells. I mean, I'm not kidding. So you have your cells. The cell needs. I mean, they are not cared after they die. So what you do, it's not a joke, it's really what happens. So you need to keep them at the right temperature, you need to keep them with the right concentration of nutrients, and you need to wash them. Which means that periodically, more or less 4 or 5 days, you go to your tissue, which is a very well confluent tissue, and you spray some water. And this reduces surface tension and addition between cells. Cells start floating and deposit again on the substrate, creating a confluent tissue again. So, yes, probably at the beginning they grew from the center, but then could be that these are images coming from cells that have been washed a couple of days. Okay. So the idea was to give a look to how can we build hexanematic hydrodynamics. And the main ingredients are going to be cellular density and velocity field, and I need to describe nematic and hexatic symmetry. And then this as a constraint. So I just want to retain these 4 elements. And once the good thing is that, okay, your equation comes relatively for 3. One is the continued equation for the density, the other one is the analysis of T. So again, I had a pressure term, which the friction term, eventually viscosity, and some stress, which accounts for the elasticity of the liquid crystal and the flow alignment of the liquid crystal. But this time, this term will actually be both for the nematic and for the hexatic liquid crystal. So these are all of these stress tensors can actually be split in a nematic and hexatic. And
I have a stupid question. How do you avoid evolving 2 Q fields?
How do I avoid?
Yeah, in other words, if you had a single symmetry, right, like in nematic, you can easily understand that the evolution of it, but now you have two symmetries.
Yes.
You bury them both in a single cube?
Absolutely no. I have two different.
All right. That makes,
And this is next.
all right, good.
But actually, this is a good point. I mean, the two symmetries are independent. The question is how the two symmetries interact. This is a way ahead. Now the point is that even to build an hydrodynamic theory for an hexatic liquid crystal, it's not trivial, and the terms don't look exactly the same. Remember now I have this object, it's not a two-rank tensor, it's a six-rank tensor. So the equation would look, the explicit expression would look pretty nicely. I'm not reporting it here, but eventually, if you write them in terms of the psi and the phase for the hexatic symmetry, the kind of look nice. So again, I have my resistance equation for the nematic field with the correlational derivative, a flow alignment term, and an acid relaxation, as described before. And the same exact terms, which would appear different, whose explicit representation would be different, but whose physical meaning is basically the same, also during the evolution of the hexatic tensor. So there is another ingredient which has to be discussed, and this is the hexatic stress active. This is something which I want to do a bit more in detail. And even in this case, I would have two contributions. One for the nematic one, the other one for the hexatic. One is something that we've already discussed before. The idea is that each swimmer, each object exerts a force in one direction at one extremity and an opposite force in the opposite direction. And I can constrain this object, and this will give rise to a term which can be written in a force density, which can be expressed as the divergence of a tensor, and this tensor is alpha 2. So my alpha 2 is actually the active stress tensor for the nematic. I need to redo this procedure for the hexatic case. So what I do is I take one object, one cell, one hexatic cell. I need the hexatic symmetry, and I can imagine that these cell exert a force at their vertices, and eventually I can write an expression for the force density for each force, for each vertex, and cross grain this expression. And what you find if you do this is that you can actually write a stress tensor which feature a fourth order derivative of the hexatic stress. So I'm basically contracting 4 of the 6 indices of the 6-rank tensor. Yes, exactly. That's exactly my reaction when I saw the equation for the first time, because it's not very nice to deal with this object, as I'm gonna tell you in a second, and to understand this. But it's actually what we were looking for, because now we have a theory, and we want it in some way to test our understanding of the theory. So what you can do is that you can linearize the brutally linearize the equation. Yeah, it's good, but sure. So the cool question is, yeah.
So Q just has Q2, and Q6 is just Q6. So there's no cross terms by symmetry.
There could be. I mean, there could be absolutely, and this would look like it's in the. You can express in the free energy. It would be a contraction of Q2 tensor 3, totally contracted in the free energy to Q6.
So why
because they are basic, they can be, they are there. The point is that the elastic term should be here. So this H2 is the molecular field, which is basically the functional derivative of the free energy with respect to Q2. So if I have a term in the free energy which makes a coupling between Q2 and Q6, you have it and it's it. It doesn't appear explicitly because this is a functional derivative.
But why there's no such a term in the GPU?
But it's there.
We wish we wrote it.
It's here inside. I mean, this.
That's the equation stress about equation, but I'm talking about the equation of motion of a cube.
Okay.
It's also there. Sure. Maybe it's there. I just need to see H2, right?
Yes. Yes. Feeling the relaxation, yes.
Earlier on, I didn't mention, the expression built up H2 is only the.
Yes.
There are other terms of H2.
Absolutely.
Do not have gradients in. And now.
That's totally correct. I mean, absolutely. So this H2 again is the functional derivative of the energy. The free energy is
It's not just that.
It's not just that. This is just absolutely. I mean,
Is that I was just seeing U2 and then.
No. It's absolutely correct.
So you have this, the reason you need 4 derivatives.
Yes.
Is because of the particular way trying to extract stress by putting these forces out of the vertex.
No, it does not depend on your orientation.
Why can't I leave the stress just by contracting U6? We might proper tensor to get a second one. I mean, it's got.
You can actually do this and you find out that this doesn't work. When you cross grain, it wouldn't work. You would have to attract, it would renormalize the track.
We do need some very. Okay.
But it actually does. I mean, the answer is that it does, and it renormalizes, but it's a renormalization. Yeah. Okay. So the idea is that we can try to linearize the equation and try to extract some information from this most gross set of the rate. So if you do it, what you find is that so the idea was to study the density fluctuations. Okay, so and the expression there gives it's basically the expression in the Fourier space of the density fluctuation in the linear system. And what you find is that basically you get 2 branches for your density fluctuation. One branch is basically increasing at large boost, so at the smaller scale, while instead the other one is decreasing at the smallest scoop, which are the large scale. And this corresponds to basically 2 possible behaviors in the 2 different regimes, so the small scales and the large scales. What you see here are basically the dashed lines are the branches from predicted with the linear stability analysis, while instead the continuous line is a direct numerical integration of the total set of equations, so not the linearized one. And they, match at least phenomenologically pretty good. Well, actually, what we did also was, I mean, this is simply characterization, and we wanted to give a look to do this spectrum of fluctuations reproduce what we see at least in the numerical models for epithelia. So what we did was to measure exactly the same. I mean, I want to stress that this rock here is a continuous object, is the hydrodynamic density. Well, instead, is the fluctuations that we measure for the model epithelia are basically the fluctuation of single cell resolved models. There are basically 2 totally different objects, at least at the phenomenological level, we still observe the kink. And what is interesting is that this kink, these two behaviors, this large scale and this small scale behavior, actually occur at the crossover point where nematic, of the predicted crossover point where nematic and hexatic symmetry switched the rings.
What's happening at large? Any theory that like use this thing?
I'm gonna get there in a second, because that's actually what I presented so far. I mean, to give a better, I mean, it's a very enough pattern twitch, to be honest. So what we did was keep studying. And one system that could actually hide a lot of information, but even from a biological perspective, is the case of channel. So one wants to understand how cells move in narrow spaces. So this is actually what, for instance, what happens during the metastatic. So we can actually characterize that. So here you see a movie of cells growing in channel. And so this is actually an experiment. So you can actually image this stuff. You can actually make measurements. And I know that Germany is working on this. And that actually, and so the idea is basically the following. We simplify the problem. We get rid of the nematic part because we want to investigate only hexatic activity, because it's the one which is least understood. Then we will, in the future, proceed to couple them. But right now, what I want to do is take a minimal model and study it. And what I can do is putting my system in a channel and impose independence in the channel direction. So the only gradient which will act are in the perpendicular direction. And I only retain among all the stresses that I had before just two terms. A term for viscous dissipation and another term for the hexatic activity object. This is in the Navier-Stokes equation, linearized version, oversimplified version. And this instead the Beres-Stephens equation, where this theta is the theta which represents the orientation of the hexatic side. And so basically this is the elastic term which leads to relaxation, and this instead is the vorticity tapping with the angle. So I mean, yeah. And you can actually extract a length scale, an active length scale, as the square root in the ratio of alpha, which is the activity, intensity of the active stress, and K, which is instead the stiffness of the liquid crystal, the elastic content. So again, what you can do is do stability analysis, and what you find is something a bit not exactly what you want, because this system equation is intrinsically active, which means that the large wave numbers, which means at the smaller scale, a small perturbation would diverge would diverge. So it would grow exponentially fast. And now we needed to fix the model. And I mean, the point is that look at this term. I mean, if you go to Fourier space, this object here, which is does not have a defined sign. This is an alpha phenomenological parameter in front of it. It could be anything, could be positive, whatever. It's grows like in Fourier space, like 2 to the 5. While instead, viscosity is dissipating like 2 squared. Normal. I mean, the rate at which activity, hexatic activity is pumping energy on that scale is technically larger than the rate at which viscosity can dissipate. So following a procedure which is standard in the context of complex fluid, we introduce an hyperviscous term. So it's still a term which has a definite behavior in Fourier space, only dissipate energy, and dissipate energy in an order at a rate which is fast enough to take out the energy which is injected back. And now, well, now
It's like
yes. Well, I mean, but everything is complex. I mean, you need to take the real part of the omega. I mean, you need to go to Fourier space. I mean, that's just an argument.
yeah, but this system to my mind is square. Why?
No, I think it puts that into the d5 and divide. So it becomes beta 6.
Yes, yes.
Oh, yes. Okay. Okay.
Okay. So if you do it, now obviously your rate of growth of the instability would be well defined at large scale. So basically you have two regimes. One regime at small scales where the system is stable, and the system, and another regime at larger scale where the system might go stable, but somehow this would be the largest in the box, the channel. So I'm going to show you what happens in these two cases. If you consider a channel box, a channel which is smaller than the typical size of the instability, what happens is that the system has an overswitch at the beginning and then is set up on a flat base. So sorry, I mean, this is the theta profile in the channel. So the orientation, how the orientation of this hexatic star changes with respect to the y direction, but instead this is the velocity. And if you instead increase the size of the channel, what you observe is that you get a modulated profile for the theta angle, while instead the velocity is positive while it is negative. And obviously, this is a system which is unstable on the largest scale, of the channel. In general, however, what you could do is something a bit more generic and decide to keep both. Go a bit faster to finish on time. So what you can do is going too fast. So what you can do is now consider both viscous term and dynamic viscous term. And this is actually opens up a lot of possibilities, because now your linear growth rate is well defined at larger scale, because this is eaten up by the 2π term, I mean the 2/6 term. And however, it's a range of scales where the system can go on. So what happens is that if you consider a channel which is smaller than the critical, this large, I mean this pussy, I mean pussy is the wave number where the system is maximally unstable. Then what happens is that the system goes to features an instability over the larger scale system. However, if you consider now a larger channel, the main, I mean, the critical size of the stability remains the same, because it's independent from the scale of the system. So this means that the scale, the two number where the system would go unstable is always that one. It's not anymore the size of the system, because it stays to C. So it does not matter if you consider a larger channel. And what happens is that in this case, you can actually observe a serial modulation of the theta profile. So one last remark. This is something that we are currently doing with the ETH, with one of my students at Koch University. So we are trying to check these picture on a model epi. So we consider a multi-phase model in a channel, and we let it go, and we measure the velocity profile of the motion of the system. And what we find is that the typical profile of the cells is actually can feature these kind of profiles. So a unidirected motion, one direction of the channel up to 50-50, and at a certain channel. Now what I'm gonna do is that I'm gonna increase the channel width, and what happens is that instead I observe several modulations. These are basically lanes along which the cells move. So somehow what happens is that you have a lane of cells moving in one direction and another lane moving in the opposite direction, which is exactly what was described by the continuous theory relation. A critical, a typical wave number for the instability, which is leading to these lane effects. And if you increase the channel with even more, what you observe is that you can observe, you find more and more peaks until at a certain point. I mean, the system will stop seeing the boundary conditions. Then you don't, when you average over time, you don't really observe any flow.
And it seems you, just on that point, you're saying you make the box arbitrarily large, you get arbitrarily large number of wiggles.
Exactly.
Right, I mean, what terminates that? I mean, you're putting a lot, you're putting a lot of energy into maybe domain boundaries.
Yeah, but it's not true anymore that the velocity goes in one direction, in one direction only. I mean, like this, the channel is pretty much confined. So you can actually neglect, in a sense, the transverse velocity of the cell. The model is a transverse velocity, but it's actually, negligible with respect to the longitudinal component. If you send the system size to infinity, at a certain point, this is not true anymore because cells can actually compress also in the transverse direction. It can create flow. So I would say, in a sense, that to give an answer, to make a guess, the critical scale would be the one for, some units of the crossover points between hexatic and nematic. But it's more like a guess from intuition. So I think. Well, that's actually the objective, right? We are trying to. This is the way you can do it. You can. Two things. One thing is that I trust the multi-phase spin model. When I first computed, actually, we compared the decay exponent for the hexatic and nematic order on both experiments and multi-phase spin model, and I was personally shocked to see that not only to observe the crossover, things that you don't see in the Voronoi model, but the experimental data and the numerical data overlap. My first reaction was like, we can't publish. They will think which. And this is something that in this, when you do simulation in soft matter, especially in active matter, you're happy if you find the exact scale. Even the points superimpose the luck in a sense. So I trust them. On top of this, I know for sure there is people doing experiment on cells in a channel, which is working exactly on this. So hopefully in some time we will.
But the movie that you were showing the way you do it.
Yes, that was experimental.
So why you cannot do it?
Well, because I'm not collaborating with them. I have the movie, but I don't have the collaboration. A very practical reason. I'm not collaborating with them. Yeah. So
that's the sociology.
Sometimes. So I'll very rapidly conclude.
What,
Yeah.
what element of this story is sensitive to these nematic hexatic coupling? You know, in other words, the different interaction of the two different Q fields in the evolution.
I would say, first of all, in my interpretation, there are basically two, nematic symmetry, which happens on the larger scale, is important for the migration of big group of cells. So the way certain layers compressed, it's nematically aligned, and this will determine how group of cells propagate
That
in
makes sense.
in space. However, if you want to understand how cells switch neighbors, so if you want to go to the minimal, or not on a particle description, because the description is not done. It's to describe the motion of the single cells. But if you want to look at the reorganization of, for instance, if you want transition, then in that case you have to look at hexatic symmetry. Or if you confine the system in a capillary, if your epithelium somehow has to, handle for a very tiny space, well, in that case, you need to consider hexatic symmetry. So this is actually work in progress. Very in progress. And so I conclude that, and so the idea is that basically active materials can, or at least some of them, be described with a hexagonal approach. And in particular, if I want to describe an active system, one of the things that I need to do is determine the symmetries of the system. If I have the symmetries, somehow the equations for the evolution of the constrained object are relatively free. And in the case of epithelia, these symmetries are the hexatic and the nematic one, and these allow us to justify the existence of some observed non-equilibrium steady states in experiments, and in the future we are gonna keep working on them. Surface of simulation to decipher and the continuous model to decipher the role of topological effects, nematic and hexatic topological effects, their interaction that we actually detail as a physical interpretation in their function to, and this is actually effect functional to many biological processes, and well, thank you for your attention. We have this one word, it's fair knowledge of the work of people. The first line is basically the three PhDs at Leiden University and my previous professor, and these are now my current collaborators, Anti-diffusive, which speaks at the University of Bari, and which, despite the fact that he's incredibly young, they are undergraduate doing an incredible job of data. And then the funding, ERC, which paid my salary in the past, is my book. And then WO, which paid for the simulations, and now ENBO, which is paid myself now, and the cluster at the Coast University, which now is thanks.
Okay. Thank you. We've got quite a few questions. More questions? Yeah.
I'm not sure if a little what you described here is
Okay, first of all, okay. Yeah. Okay, first of all, the first case, epithelial tissues are usually monolectures.
So they don't really exist. I mean, there are some exceptions, but they don't exist in body. Example, skin, or what covers the villi in the intestine. They're usually used in the animal body, any animal body, whatever, as a protective layer to separate two different biological environments. So they can bend in 3D, but still be two-dimensional. Now, what happens, it's a question which interests me really a lot, and I stay up at night for hours speaking about what happens to liquid crystal on close surfaces. But what happens is that you are changing the topology. So the first things that would happen, let's take the easiest case, the case of a sphere. If you try to comb an airy ball, then you would never be able to eliminate that topological defect. You are always left with a +2 topological charge. So what happens is that basically, if you have a polar object, then you would have two defects of charge, one +1 and +1 at the North Pole and the South Pole. And it doesn't matter, it's a matter of topology. So it doesn't matter if it is, compressed or whatever. I mean, as long as as long as you can deform the boundary continuously to a sphere, that's the total charge that you can get there. So you're actually, that's a way, which is called topological confinement, to position defects to place defects in a system, because they're stable. Defects are usually answered and washed out by elastic processes. I mean, through the annihilate, they can, but they on a sphere they mathematical stuff. So the question is, what happens? Sorry, probably I'm over answering now, but the point is that if you have an hexatic liquid crystal, you would have the football. You need at least 5 pentagons to shoot the ball, right? So those are the defects in an hexatic pattern. And you can't eliminate them. So those defects would stay if you put an hexatic liquid crystal on a sphere. And if you want to see an embryo as an hexatic liquid crystal, the role of topological defects would be extremely important. One more thing, there is a lot of evidence that defects plays a huge role in the roughening of surfaces. If you have a topological defect, this usually in biological systems gives life to protrusion. There are a lot of examples. And the extrusion of cells in epithelial tissue is just one of the examples. And what happens in that system is that the roughening gives rise to invagination. My understanding is that the presence of hexatic topological defects, which usually actually occur scarce, so regions where there are densely populated on lines of defects one in a row, is playing a lot in the dynamical process of invagination of food.
My very first that your thought
It's an interesting question, but I would honestly say that I see a lot of separation between the current understanding. These are the easiest thing that you can do, simulations on a flat substrate. So we would enter the regime, the realm of speculation. I love to speculate, but I would say that from a physical perspective, the first thing that I would say is that enters into the friction. We have a friction term in the equation, which is the interaction of the flow with the outer wall. I have a two-dimensional fluidic layer, and the outer wall is actually described by friction term, and minus lambda v in the
It's not just that. It's, for example, the argument that the surface is closed. It's based on the fact that it's closed. Boundaries, curvature, values, this, the surface is closed. It's not just.
I totally agree. I mean,
I agree with you, it's more complicated than that, and this is the first step that should be done, but the boundary conditions.
I totally agree.
I agree, it's a good time to stop.
You consider, I mean, just thinking of a wound, right? The possibility of the boundary condition exerting a stress,
Exactly.
then surely it matters.
Exactly. Exactly. In that case, I would say that what's playing an important role is actually the isotropic pressure. So you have a gradient of pressure. Here you have outer wall that can apply on the layer an isotropic pressure, that atmospheric pressure if you want. And instead inside you have the stress coming from what is happening in the cellular level, at the sense level. And what's happening is actually very similar to, the cells are literally able to perceive the fact that they're not confident on one side and they want to find the available space. I mean, these are a lot of interesting questions. I mean, which are, however, we are at a stage where we are happy with the, no, we are not happy, but we are looking forward to continue, but there's a lot to do.
Okay. Very interesting.